Kia ora and talofa everyone. My name is Aleluia. I'm a PhD student from Victoria University of Wellington here in New Zealand. My overall project is about the effects of ocean acidification and ocean warming on Caulerpa species. I'm originally from Samoa, a small island from the South Pacific, and in Samoa and in most parts of the Pacific, Caulerpa is edible. So it is already an important food source and a subsistence commodity in the communities. So generally from where I'm from, Caulerpa is an important fishery because of its cultural, traditional, social and potential economic values. So what I'm interested in is the responses of Caulerpa to climate change. And so the idea originally was for me to come to New Zealand and study Caulerpa in terms of their physiological responses. And then toward the end of my thesis, take all of this to Samoa and do similar tests there. However, because of the pandemic, I can't do that anymore, obviously. So instead, what I'm doing is assess just the responses to climate change in terms of two singular experiments, where we assessed how Caulerpa respond to ocean acidification, ocean warming, and marine heat waves, for example. But we also do run other experiments beyond this that assess how all of these interact. But for today, I'm just going to present to you the results of one of these two single factor type of experiments, and that will be the ocean acidification experiment. Under ocean acidification, this is what we expect to occur in New Zealand and globally in terms of the carbonate chemistry of seawater. As shown by the top plot, the red line is the increase in atmospheric CO2, and dark blue dots show the CO2 concentration in the seawater is also increasing over time. As a result, pH is already declining, as shown by the lower plot in light blue color dots. We also know that by the end of the century, we are expecting seawater CO2 concentrations that are even much higher than what is currently happening. But there are other changes in carbonate chemistry that are occurring. This includes increased hydrogen ions concentrations causing pH to decline, while at the same time bicarbonate is increasing while carbonate ions are decreasing as indicated by green dots. So already we see the immediate direct negative implications of ocean acidification on calcium carbonate organisms, including calcareous macroalgae. And what's interesting is that people have found previously that some species of fleshy seaweeds or non-calcareous seaweed are impacted by ocean acidification. And the way they found that is from natural CO2 vent sites, where species that are under this green line can benefit from increasing CO2 concentrations and tend to increase in abundance than the ones above it. So majority of macroalgae can also use bicarbonate. We call those species that could use bicarbonate CCM species because they have what is called a CO2 concentrating mechanism, which allows them to actively uptake bicarbonate. So generally, if they do not have a CCM, they are restricted to CO2, and if they have a CCM, it means they could take up bicarbonate actively, so we would assume that there wouldn't be as large a positive effect. So to identify if a seaweed has a CCM or not is by measuring their carbon stable isotope values, like how it's shown here on this figure. So as the isotopic value of the seaweed decreases, it indicates increasing use of CO2 and decreasing use of bicarbonate. Those species below this green line roughly are what we call non-CCM species, and they are entirely restricted by CO2. Species close to -10 will be using just bicarbonate, and as they move closer to -30, it indicates higher CO2 use. In most species of seaweeds within the same genus, they either do one or the other, but Caulerpa is really interesting because it is one of the rare genus where within the Caulerpa genus, it has species that could either have or do not have a CCM. So here in New Zealand, in Wellington specifically, we have these two species of Caulerpa seaweed that have different carbon physiologies. So we predict that Caulerpa brownii, which does not have a CCM, will be more positively affected by ocean acidification than Caulerpa geminata that has a CCM. But this remains untested. So what we did was we ran an experiment to test how ocean acidification would impact them. So the hypothesis to be tested here is that non-CCM species, that is Caulerpa brownii, will respond more positively to ocean acidification, and CCM species, that is Caulerpa geminata, will not respond. I also assume here that carbon stable isotopic values of Caulerpa geminata could decrease, and that is as indication for downregulating of their CCM, for example. So in order to evaluate this hypothesis, we are assessing a range of physiological responses, including a range of photophysiological responses. For today, though, I'm just going to discuss the growth responses and isotope responses. For the purposes of today's talk, I'm not going to go into the method so much. So for the ocean acidification experiments, we had 4 mean pH treatments that fluctuated around the mean pH in a full factorial experimental design. The 4 different colors you see here demonstrate 4 different mean pH treatment levels. That is 8.03 pH is also our ambient treatment. We had 7.93, 7.83, and 7.63, which is also our future extreme treatment. This is the nuts and bolts of what we had. We had 12 replicate tanks for each of our 4 pH treatments. For ease of reference for the results slides, I will always have the plots for non-CCM species, Caulerpa brownii, on the left of the panel, and geminata, which is the CCM species, will always be on the right. I will also always have the order of pH treatments from ambient on the left to most extreme treatments on the right. So for example, ambient is the blue color bar, and the strongest or extreme treatments is in red. So this is the results for relative growth responses. This is what we found. And this is really interesting because the variability is increasing in both of the species. But in Caulerpa geminata, which is the CCM species, they respond more negatively, whereas the non-CCM, Caulerpa brownii, tended to increase its mean growth rate and responds more positively. But none of these relationships are significant because there's a huge amount of variability, in particular in the more OA treatments, so that's in the lower pH treatments. What we think is happening on the left is that brownii is receiving a benefit in terms of more CO2. Geminata might be receiving some benefits from higher CO2, but in general there is negative impact on both populations. Now what's interesting is that whilst it wasn't a hypothesis of ours at the start, what we found in past literature is that, for example, for calcareous red seaweed, their internal pH changes due to ocean acidification. Like I said before, there's different components of carbonate chemistry changing, and actually we know that for coralline algae, for example, which function much differently, but we know their internal pH is altered by ocean acidification, and it's likely that the species here are also impacted negatively by OA in some instances. We think these internal pH changes are having negative consequences for some of the individuals and not for others, so that's what we think is occurring. In this slide, it shows the carbon stable isotope values. So what would occur here? If you had a non-CCM species, that is Caulerpa brownii, you would expect if they are using just CO2 already, their isotopic values are not going to change, right? However, for the CCM species, that is Caulerpa geminata, if they start using more CO2, this will be shown in their isotopic values as it will start to go towards the -30 line. We're measuring a range of photophysiological responses, for example, and I don't have the time today to go into all of those. And we are also in the process of conducting proteomics responses as well. And hopefully I'll be able to share those with you in a future day. But today, this is all I'm able to discuss with you. All right, for discussion, for growth response, our results show that relative growth response variability is large. And even though there was no significant effect, the fact that it was increasing variability so much was a strong effect in itself. For the CCM species, that is Caulerpa geminata, they got more CO2, so they can fulfill more of their carbon budget with just CO2 rather than actively pump it up, or maybe some sort of pump shuts off, which is more likely active pump senses the CO2 and then stops. And that explains to some extent the variability here. For isotope results, the CCM and non-CCM cutoff points varies for some individuals and shows that there's something going on there where there are different compounds they possess, which can also increase the isotopic value. Whereas for Caulerpa brownii, the non-CCM species, it looks really clear that they're going to exclusive CO2 use. So overall, we demonstrate from this experiment that dissolved inorganic carbon use alone doesn't predict responses to ocean acidification. And with that, I end my presentation. Thank you.